Welcome everyone to the 5th webinar of the International Eosinophil Society. This month we're back to biology. We will be discussing the interactions that eosinophils entertain with their environment and more specifically with non-immune cells. Before introducing today's moderator and reviewing a few housekeeping items, I'd like to take a look at our society's missions together. These webinars were designed as a contingency plan when it became apparent that we would be unable to hold our biennial meeting this year because of the COVID-19 pandemic. The topics and speakers have been excellent and we have had great attendance. So we are quite pleased to say that we have continued to fulfill these important missions and have been able to satisfy, at least in part, all those interested in our dear eosinophil. Now for the logistics of this webinar. If you have any questions during the meeting, you can enter them at any time in the question and answer section of this platform. Our moderator will make sure your questions are addressed at the end of each presentation. This webinar will be recorded, so you will be able to review it again and to share it with your colleagues if they were not able to attend today. You just need to go to the Society's webpage where you will find a link for each webinar we have had so far. I will now leave you with our moderator, Dr. Hirohito Kita, Professor of Medicine and Immunology at the Mayo Clinic. He is well known for his work in allergic disease, including both the early phases of inflammation initiated at mucosal surfaces and the components of more chronic disease phases. Today's presentations are right up his street. I hope you will enjoy them.
Okay, good morning, everybody, and thank you for joining us. We have 4 outstanding speakers today. And the first speaker is Dr. Seema S. She's a professor of pediatrics and medicine at the University of California, San Diego. She's also endowed chair in pediatrics. She directs the eosinophil gastrointestinal disorder clinic at the Raleigh Children's Hospital at San Diego. She's a leader in the field of the pediatric EOE, especially about the mechanism of the remodeling. The title of her talk today is Modulating the Interaction Between Eosinophils and Fibroblasts. Dr. Aceves.
Thank you very much. And my screen shared so you can all see the slides. I'll go back to the beginning here. It's a pleasure to be here this morning and talk to you. The title of my talk is Modulating the Interaction Between Eosinophils and Fibroblasts, and I'm specifically going to talk about EOE. My disclosures are listed here. They're not relevant to this talk. Just so that we're all on the same page about EOE, this is a clinicopathologic diagnosis with symptoms of esophageal dysfunction, and then you have to have an eosinophilia of the esophagus of at least 15 eosinophils per high parafield, and then you can potentially diagnose EOE. EOE is instigated by allergens, both inhaled and ingested, that induces an inflammation of the esophagus that's quite robust and of course involves everyone's favorite cell, the eosinophil, along with T cells, mast cells, and ILC2s. The disease is chronic and in its perpetuation stage goes on to have esophageal remodeling that, much like asthma, associates with fibrosis, smooth muscle hypertrophy, and angiogenesis. That remodeling can also play a role in perpetuating the inflammation, and that's where we're going to touch on the fibroblast-eosinophil interaction today. Histologically, there's epithelial as well as subepithelial remodeling. In EOE, you can see a significant amount of basal zone hyperplasia. This is very much a disease of epithelial dysfunction. Eosinophils that infiltrate that epithelium. You can get desquamation as well as dilated intercellular spaces. And in the lamina propria, you can get a significant amount of fibrosis. That leads to symptoms and remodeling in the patients with esophageal strictures that can lead to food impactions like this piece of chicken in the esophagus. Children will complain of anorexia, early satiety, they'll have reflux symptoms, and they sometimes will have trouble gaining weight. EOE is very much a disease that walks to strictures, especially if it remains untreated. While it starts in an inflammatory phase, it eventually ends up in a mixed and then fibrotic stage, and there are endotypes that have been associated and shown to be mechanistically involved in the progression or certainly with phenotypes of this disease. So I'm going to start by telling you a little bit about the role of TGF-beta in EOE because it pertains to what we're going to talk about in terms of fibroblast and eosinophil interactions. So our lab and others have demonstrated that TGF-beta is significantly expressed at the protein level in the EOE esophagus. It's made by a number of cells, including eosinophils that you can see on a double label with MBP and TGF-beta, by mast cells that are shown here on a double label with tryptase and TGF-beta, and also in the epithelium. Of course, TGF-beta then talks back to those cells to create a network. States that give you increased TGF-beta expression, there's a SNP in the TGF-beta1 promoter that creates a naturally occurring state in humans of increased TGF-beta1 expression in the esophagus, can lead to histologic remodeling. So children that have a genotype of TT at the C-509T promoter have increased numbers of TGF-beta1 positive cells that you can see here. They also have increased numbers of mast cells that make TGF-beta. And if they happen to be food sensitized, they tend to be fibrotic. So in the esophagus, TGF-beta1 induces myofibroblasts, and you can see that with these yellow cells that infiltrate here. And more recently, we've shown that the extracellular matrix that's derived from a fibroblast of a patient that has severe EOE, when you put a normal fibroblast on top of that EOE matrix, can actually alter the function of the normal fibroblast through proteins like thrombospondin 1. And if you utilize that system of an EOE extracellular matrix with a normal fibroblast and you block with the TGF-beta chemical inhibitor for its main receptor, TGF-beta-R1 or ALK5, you can block these inductions of collagen and alpha smooth muscle actin. So we know that TGF-beta seems to be involved in the remodeling that occurs in EOE. So TGF-beta can interact with a number of things, and we became very interested in collaborating with Mick Croft's lab, and all of the data I'm going to show you has been a collaborative effort between Mick's lab and mine. And we became interested in how the TNF superfamily members could interact with TGF-beta and act on their own to induce changes in EOE. So light, which is TNF SF14, has pleiotropic effects in remodeling and inflammation, and there is a vast amount of literature that I don't have time to show you all of, but I can tell you that light affects structural cells, including epithelial cells and fibroblasts and smooth muscle cells, to change the way they produce ECM and chemokines and cytokines. So we're going to talk a lot about light, or TNF SF14. Light is made by activated T cells and B cells, and if you concentrate on the right-hand side of this panel, what you can see is that light can interact with TGF-beta1 to change the function of fibroblasts. So we first asked whether light receptors were expressed in EOE, and we did this by looking at RNA sequencing data. EGIT Express is a publicly available data set from Mark Rothenberg, and if you look in EGIT Express, you can find that both of light's receptors, LT-beta-R and TNF-R-SF14, which I'm going to refer to as HVEM from now on, are both significantly increased during active EOE. We then looked in esophageal fibroblasts, and what we found was that in active EOE fibroblasts, so these are derived from a biopsy from a child that has active EOE, there is significantly more HVEM expressed on those fibroblasts as opposed to fibroblasts that are derived from a normal esophagus. When we looked at LT-beta-R, there's actually more LT-beta-R than there is HVEM, especially in the normal state, but there's not a lot of difference between EOE and normal biopsy fibroblast. We then asked whether light itself was expressed in EOE, and you can see by RNA sequencing using EGIT Express, light is indeed significantly induced during the active EOE state. We then asked specifically if the T cells in the esophagus during active EOE were making light. And we did this using single cell sequencing data that was generated by Ting Wen and Mark Rothenberg. When we assessed whether light was being expressed in the T cells, what we found was that light was expressed significantly across multiple of the T cell subtypes that are found in the esophagus. And that it was also induced in this T8 subset, which is known to be the pathogenic subset in the EOE active esophagus. And when we looked at the number of transcripts for light as compared to IL-5 or IL-13, which we of course know are pivotal for eosinophils in EOE, we found that light was expressed to a significant amount, almost as much as IL-5 and IL-13. We then looked to see if light was being expressed at the protein level, and we did staining of EOE active biopsies, and that's all I'm showing you here. And then we quantified how many light-expressing cells there were. And you can see that during the active EOE state, there's significantly more light expressed both in the epithelium, and there's also a trend towards having more light-positive cells in the LP. So we were convinced that this TNF family member was induced in EOE. So then we treated fibroblasts with either TGF-beta1 or with light to see what light could do to a fibroblast. They did have very distinct profiles of transcriptomics. And while TGF-beta induced a myofibroblast phenotype, which was expected, light induced an inflammatory fibroblast within within the esophageal fibroblasts. And it was interesting to us that in ulcerative colitis, there's a subset of inflammatory fibroblasts that have been found on single-cell sequencing that express a number of proteins and transcripts that are also induced in the presence of light. And those include TNF, SF13B, which is BAF, C3, CD74, IL-32, and ICAM1. So we then looked between healthy and active EOE fibroblasts to see if there were differences in these transcript levels and whether they were induced by light. What we found was whether you were a healthy fibroblast from the esophagus or an EOE fibroblast from the esophagus, light would induce this inflammatory transcriptome with the induction of ICAM1, IL-32, and IL-33, although this was higher at baseline in normal esophageal fibroblasts. And we're going to focus on ICAM1. So then, because we wanted to know the interactions between TGF-beta and light, we asked whether TGF-beta would actually alter the expression of light receptors. When we looked at the effects of TGF-beta1 on LT-beta-R, we didn't see that there was much induction. So the red and orange here are the isotypes, and the blue is vehicle, and then the green is the TGF-beta treated cell on FACS analysis. On the other hand, what you can see is that HVEM is significantly induced in fibroblasts that are treated with TGF-beta. And if we look at a number of patients, each dot here represents a different person's fibroblast, we can see that TGF-beta induces HVEM both on healthy and active EOE fibroblasts. So based on that, we then asked whether light and TGF-beta could work together to increase the expression of ICAM1 or other modulators that could alter the ability of fibroblasts to call in inflammation, since this was an inflammatory phenotype of the fibroblast that seemed to be induced in the presence of light. When we looked at the expression of ICAM1 in the presence of both TGF-beta1 and light, what we saw was that it was significantly more ICAM1 transcript when you had both TGF-beta1 and light present. We found the same thing for some inflammatory markers like IL-32. And while the effects were a little more diverse from person to person, we also saw that there could be interactions on actin genes as well as procontractility genes such as phospholamban. So then we asked about differential effects of light and TGF-beta1 on adhesion molecules, and we focused on ICAM1 and VCAM1. What we found was that light could induce ICAM1, but that it was improved when we added TGF-beta to that, and that is, we believe, probably because we're inducing HVEM. On the other hand, VCAM had differential effects such that the addition of TGF-beta1 and light actually brought the VCAM levels that were induced by light back down. So you can see that these molecules interact differently on esophageal fibroblasts to change their adhesion molecule profile. You're looking here at FACS analysis of cells that were treated with light, TGF-beta1 or both. And what you can see is in the presence of TGF-beta1 and light, you get an SMA positive, ICAM1 positive cell shown in quadrant 2, that when you stain, sure enough, you can see that the cell is now expressing ICAM1 as well as smooth muscle actin. On the other hand, when it comes to VCAM1, and if you look in the ICAM1 gate, we find cells that are triple positive for smooth muscle actin, VCAM, and ICAM1, and also cells that are negative for VCAM1 and only expressing ICAM1, so falling more in the category of the cell in the middle panel. So then we asked, okay, ICAM is on and light and TGF-beta induce that. So what happens to the eosinophil and can we get it to tether? We worked with Praveen Kuthoda's lab and we got human eosinophils from Praveen's group, and we incubated those eosinophils with esophageal fibroblasts, and we treated them with light or with vehicle. And what you can see, which is really just so cute, is that when you treat with light, the eosinophils start to tether onto these fibroblasts. They do tether at baseline, but there's a lot more when you add light. And they tether, and they also form these little clusters of groups of eosinophils that are now hanging onto the fibroblast. If you stain with EPX, you can see the same thing, and we think that maybe the eosinophil is being activated. You can see some extracellular stain here, and it's something that we're very interested in understanding better. If you quantify the amount of clusters of eosinophils per fibroblast in the presence of light, there are significantly more eosinophil clusters when you treat with light. And then Mario Manresa, who did all of the work that I'm showing you today, then treated with an ICAM blocking antibody to see if we could block the interaction between the eosinophil and the fibroblast. So you can see if you add light and you use a monoclonal antibody that blocks ICAM1, you break up the eosinophil fibroblast partnership that was occurring. So we believe that ICAM1 is involved in the interaction between an inflammatory fibroblast and an eosinophil. We then asked whether we could find these types of couplets in vivo. We stained with alpha SMA as well as Vimentin to show that there were double positive cells, which you can see here in yellow, in the active EOE esophagus. And then we asked if those cells could live near an eosinophil, and we used an EPX stain for that. And you can see that these Vimentin positive cells can be found next to the eosinophil. So we do think that this is relevant in vivo in a person. Mario's gone on to show using RNA-scope, which is what you're looking at here, that transcripts for ICAM1 as well as Vimentin can co-localize in the active EOE esophagus. And so RNA-scope is really lovely, and every little dot you're looking at here in orange represents a transcript for a Vimentin molecule, and every green little dot represents a transcript for ICAM1. And what you can see when you get to high power is that these do tend to cluster together. They can be found separately as well as also together, showing that the same cell is capable of making both molecules, both transcripts in the same cell. Mario's gone on to ask which of the receptors could be the most relevant for the interaction between these eosinophils and the fibroblasts. And this is work that he presented at the DDW just recently. So in this case, you're looking at an H&A stain of fibroblasts and eosinophils that have been incubated together, either in the presence of a non-targeting siRNA and vehicle. And you can see again that you have these clusters and they do adhere to some extent at baseline. And that's not surprising since we do have ICAM production at baseline within the fibroblast. When we add light in the presence of a non-targeting siRNA, you can see what we saw before, that the eosinophils are now clustering together and adhering to these fibroblasts. If we add a blocking, not a blocking antibody, but an siRNA to HVEM in the presence of light, you can now appreciate that there are significantly fewer eosinophils tethering to these fibroblasts. And interestingly, when we add a siRNA for LT beta R, we also see this decrease of eosinophils tethering to the fibroblasts in the presence of light. If you quantify that out, you can see that in the presence of light alone, with a non-blocking siRNA, that there is an increase in the amount of eosinophil fibroblast clusters, but in the presence of either a blocking siRNA for HVEM or for LT beta R, you get a decrease in this interaction between the fibroblast and the eosinophil. We've also asked what type of signaling pathways, whether HVEM and LT beta R would blockade would change the amount of ICAM1 that's being expressed, because we think that that would be the mechanism by which this eosinophil would be blocked from tethering to the fibroblast. So here you're looking at quantification. Every different color is a different person's fibroblast. And what you can see is that when you add light in the presence of a non-targeting siRNA, that you get ICAM1 production. However, if you add a blocking RNA for HVEM, or in the presence of light, you get a decrease in the amount of ICAM that's being produced. And if you add blocking siRNA for LT beta R in the presence of light, you also see a decrease in the amount of ICAM1 produced. So it appears that both HVEM and LT beta R are required for ICAM1 induction and also for the tethering of the eosinophil to the fibroblast in our system. Mara has also gone on to begin to understand the signals through NF-kappa B that can be involved in this, and which receptors, and this is a work in progress right now. And you're looking here at ICAM expression and using BAY1, which inhibits the canonical pathway, versus NIC, which inhibits the non-canonical P52, P100 pathway. What we find is that, once again, if you treat with light, you have an increase in ICAM1, utilizing either a canonical chemical blocker of the signaling pathway for NF-kappa B or using a non-canonical inhibitor. Both states will decrease the amount of ICAM1 produced. So I'm going to try and sum that all up for you to tell you that we think that light is expressed by T cells, especially activated T cells in the EOE esophagus. Light, by binding to its receptors, HVEM and LT beta R, can induce the expression of ICAM1 on the esophageal fibroblast. In fact, we see more ICAM1 in the active EOE fibroblast. TGF-beta1 that is made by a number of cells within the esophagus, including the eosinophil, the epithelium, and the mast cell, can then bind to its receptors, increase the expression of HVEM, interact with light to further increase the expression of ICAM1. And at the end of the day, we believe that that allows the eosinophil to come in and tether to this inflammatory fibroblast. So in conclusion, EOE is a chronic disease that, of course, involves the favorite cell here, the eosinophil. We find that TGF-beta1 is increased in EOE and believe that it's involved in the remodeling process that occurs. We've shown that activated T cells in the EOE esophagus express light, and that light drives an inflammatory phenotype of this fibroblast that's not just transcriptional, but also functional. If we drop the levels of HVEM or LT beta R, we decrease the amount of ICAM, and we also decrease the amounts of tethering of the eosinophil via ICAM to the fibroblasts. So I just want to thank you for letting me come talk to you today about all of this. I want to thank my collaborators, Mick Croft, who is a vast bastion of knowledge on all of the TNF family members, Nate Lewis, whose lab did all of the bioinformatics for us, and Praveen Akuthoda, who you all know and also love, I imagine, as much as you love the eosinophil, who gave us the human eosinophils for our tethering assay. Mario Manresa is a postdoctoral fellow in our lab, who's now actually a project scientist in our lab. And we're very happy to have him, and he has really done the lion's share of this work. Thank you very much.
Thank you, Seema, very much. Wonderful presentation. There was in the comments, there is one comment about thanking you for the shedding light on eosinophil functions.
Why, thank you.
So other questions include what kind of eosinophil receptors are being used to interact with? Do we have a chance to look at?
So that is a wonderful question. I mean, we're assuming that it's like LFA1, MAC1, but we have not looked at that, and I think that's a really important question. Which receptors on the eosinophil are important not only for it binding, but how is it being activated, and is it degranulating? And maybe next time I talk to you, I'll have an answer for that, but I don't right now.
Yeah, we could speculate like a beta-2 integrin, especially like alpha M beta-2 like molecules. Yeah, very interesting to see how which molecule is being used. Another question is there any kind of correlation between the clinical severity or symptoms of the patient and TGF-beta or light expression?
Yeah, so that I think is a really important question for people that have EOE. And so we've shown that TGF-beta, the TGF-beta SNP can associate with histologic severity in patients that have EOE, at least children that have EOE. In terms of light, we haven't looked at things like SNPs. What I can tell you is the active fibroblasts that we've used in all of our studies tend to come from pretty severe patients who don't respond to therapy. And we're in the process of looking at receptor expression by disease severity in EOE. And my suspicion is that light and its receptors will be more involved when you have a more severe phenotype. I mean, light is kind of this cool molecule that allows you to cross the bridge between inflammatory and fibrotic disease, and maybe, really at play during that transition from an inflammatory to a fibrotic, inflammatory mixed phenotype to sort of that end fibrotic phenotype. But yeah, it's a great question. I think we have inklings into that, but don't have definitive answers yet.
Okay, yeah, thank you very much. We have several other questions, but I think for the sake of time, I think we have to kind of stop here. And Seema, if you can go to the Q&A and
Sure.
Address several questions we couldn't, and I would appreciate that.
Of course, yes, I'm happy to do that. Thank you so much.
Yeah, thank you very much. So I'd like to introduce next speakers. The next speaker is Dr. Allison Fryer and Dr. David Jacoby. Dr. Fryer is a professor of medicine, and she's also associate dean for graduate studies in Oregon Health and Science University. Dr. Jacoby is a professor of medicine, and he's a chair of medicine at the same institution. And probably, as you know, Dr. Fryer and Dr. Jacoby have been working on neuroimmune interactions in eosinophils in the lungs over 35 years. So it has been very long collaboration, and congratulations. Hopefully every time everything goes very well. So I think this time I'd like to ask them to present their works, and the title is Eosinophils Change Airway Nerve Structure and Function in Asthma. Dr. Fryer and Dr. Jacoby.
Thank you so much, Hirohito. Let me see if I can share my screen. Okay, so thank you. The title of our talk today is that eosinophils change airway nerve structure and function in asthma. David and I are both funded by the NIH. We have no conflicts of interest. What you're looking at here on the right-hand side is a whole mouse lung that has been stained. So you can see the trachea and the bronchi and the bronchioles in blue. Asthma is a disease that is characterized by chronic inflammation, and it is often characterized by the presence of eosinophils, an abnormally high number of eosinophils, and airway hyperactivity. So there are bouts of periods where the lung is quiescent, and then periods where the lungs bronchoconstrict, so the airways get very narrow and it makes it very hard to breathe, or you have that characteristic wheezing sound. What I really want you to see, though, in this picture of the whole mouse lung are all of those little like lines all over the blue airways and the trachea. Those are all nerves. And so what I want to really illustrate is that the nerves are the lungs receive a very, very rich nerve supply. And the talk today will be showing you the evidence where eosinophils change the function of the nerves in the lung and contribute to asthma. It's been known for a long time that airway hyperactivity in the lungs is actually mediated by the airway nerves. This is data in a mouse. If you look at the left-hand side, if you give a mouse serotonin by inhalation, you'll get a very small bronchoconstriction, and that is the open circles and black lines that are marked healthy. If you repeat that experiment in an animal that's been exposed to antigen, the bronchoconstriction is much, much higher, and that's the purple line marked with antigen. So antigen causes airway hyperactivity in mice. But if you cut the vagus nerves on the right-hand side, that difference goes away. So this shows that airway hyperactivity is mediated by the airway nerves. We can repeat these experiments in dogs and also in humans. Of course, not cutting the vagus nerve, but by giving pharmacologic drugs that block the neurotransmitters from the nerves, you can prevent airway hyperactivity. So what do the nerves look like? And I'm going to give you a quick overview of airway nerve anatomy. There are two branches to the nervous system that are really important in the lungs. The first is the parasympathetic, which is one of the autonomic nervous system pathways. And so that's on the left-hand side. This is an illustration given to me by David Baker from UCSF. It's a ferret trachea, and the top is where the larynx is, and the bottom would be the bifurcation into the two bronchi. And I think you can see all of those black lines are nerves. The parasympathetic nerves have their origin in the base of the brain, and then they run to the lungs through the vagus nerve, which is that very thick black line marked with a V. They then branch from the vagus and supply the airway smooth muscle. Along the way, there are parasympathetic ganglia, which are clusters of nerve cell bodies. Katie Liebold has provided an illustration of what one of those ganglia might look like. The parasympathetic nerves provide the dominant autonomic control of the lungs. They provide airway tone, and when they release their neurotransmitter, which is acetylcholine, the airways constrict and get narrow. The other nervous system that is really important is the sensory nervous system. And these are the nerves that line the inside of the lungs and the airway epithelium. When you inhale an irritant, it is the sensory nerves that detect that irritant, and then they fire and trigger a reflex bronchoconstriction. And that reflex is shown here. So taking you from the left to the right, the sensory nerves line the inside of the lungs. If you inhale an irritant, you can see that black line, the sensory nerves fire, that signal transfers to the brain and then back down through the parasympathetic nerves, which are shown in red. And then the parasympathetic nerves release acetylcholine and cause bronchoconstriction. So we can stimulate a reflex bronchoconstriction by directly stimulating those sensory nerves with serotonin. We can also stimulate the parasympathetic nerves by electrically stimulating the vagus. And so these arrows really just illustrate how that reflex works. And now I'm going to focus on that little bit of area where it's got a blue circle, just looking at the nerve ending. So one of the key ways in which eosinophils change the parasympathetic nerves is that they can actually increase the release of acetylcholine from parasympathetic nerve endings. And they do that by interacting with M2 muscarinic receptors. So this is that nerve ending that I mentioned on the parasympathetic nerve side, so autonomic nerves. The release of acetylcholine is normally inhibited by M2 muscarinic receptors, and those are shown in the red squares. Acetylcholine will then bind to M3 muscarinic receptors on airway smooth muscle. The M3 are in the yellow sort of rectangles, and that causes the smooth muscle to contract and the airways to narrow. So in a normal individual, the amount of acetylcholine is tightly controlled by those M2 muscarinic receptors. However, in an animal that is antigen challenged, we know those M2 muscarinic receptors no longer function properly, so there's too much release of acetylcholine. Same is true in humans with asthma. The M2 muscarinic receptors are not functioning properly. So the question is, what is changing the function of the M2 muscarinic receptors? And one of our earlier observations was that eosinophils are associated with airway nerves in asthma. These are tissue sections from patients who died from fatal asthma. They were provided by Dr. Jerry Gleich and analyzed by Richard Costello in our laboratory. The nerves are stained in black, and then the eosinophils are stained for a major basic protein, and they are red. And so you can see at the top, this is a nerve fiber running through airway smooth muscle, and the eosinophils line up along those nerve fibers. At the bottom left, this is a nerve bundle in the smooth muscle. And again, you can see eosinophils are clustered around that nerve bundle. And then on the bottom right is a parasympathetic ganglia. And again, there are two eosinophils tightly embedded within that parasympathetic ganglia. So we are showing that eosinophils are associated with airway nerves. And in fact, there are more eosinophils around airway nerves than there are anywhere else in the lungs of patients with asthma. We're able to quantify this now using three-dimensional analysis. And so you're looking at a section that was provided by Alexandra Pincus, who is a graduate student in our laboratory. This is from an antigen-challenged mouse, and she has stained all the nerves red with an antibody to PGP 9.5. So you're looking at a ganglia, which is sort of that cluster of balloon-shaped cells in the middle, and then you can see all of the nerve fibers in red around this. All of the green cells are eosinophils, and they are stained with, they're eosinophils stained with green fluorescent protein. We can look at this in 3 dimensions and analyze the nerves and analyze the eosinophils, and it looks a little bit like this. So we use Imara's software to identify the nerves. Again, this is a 3-dimensional analysis, and you can see the nerves are now in red and are coded in red, and I think you can sort of appreciate the 3-dimensionality of it in these images. Eosinophils are in green, and we can measure the distance between eosinophils and nerves. So in the top right, you'll see that there are eosinophils around all of those nerves in that mesh of airway nerves. And at the bottom left, there's an eosinophil actually adhering to a nerve. We can quantify this in antigen-challenged mice. And you'll see that starting from the left, there's an increase in total eosinophils in these animals, but there is a significant increase in the percent of eosinophils touching airway nerves following antigen challenge, and that's the bar graph in the middle. If you actually measure the distance of an eosinophil to a nerve, and that's on the right-hand side, the dotted line are saline-treated animals, and you can see that there are some eosinophils touching nerves. So the closer an eosinophil is to a nerve, the further to the left on that graph. But in an antigen-challenged animal with house dust mite, we have significantly shifted that curve to the left, showing that there are more eosinophils around the nerves and more eosinophils that are closer to the nerves with antigen challenge. So we have strong evidence that eosinophils are recruited to airway nerves. They're actively recruited to airway nerves, and they shift and even touch nerves in antigen challenged animals, both guinea pigs and mice. So this is a summary of work that we've conducted over 20 years. And so the story for the parasympathetic nervous system is this. The parasympathetic nerves release eotaxin, and eotaxin actively recruits eosinophils to those airway nerves through interactions with the receptor CCR3. Eosinophils can actively adhere to parasympathetic nerves through an interaction with CD18 and ICAM. And then they release eosinophil major basic protein, which are all of those red circles. And we showed that eosinophil major basic protein is an endogenous antagonist for M2 muscarinic receptors. It does not bind to the M3 muscarinic receptors. So the eosinophils carry an antagonist to the M2 receptors. When you lose M2 receptor function, you have too much acetylcholine release and then too much airway narrowing and bronchoconstriction. If we interfere in any way with this pathway, either by depleting eosinophils with antibodies to IL-5 or VLA4, by blocking eotaxin with a CCR3 antagonist, or by removing major basic protein with an antibody to major basic protein, or with heparin, which will precipitate major basic protein, all of these treatments will protect the M2 muscarinic receptor and prevent airway hyperreactivity. So this data really shows that the airway hyperreactivity mediated by the parasympathetic nervous system is due to a very specific interaction of eosinophils and airway nerves. So one of the questions we've had, though, is that when we stimulate the nerves, we end up with sort of a mixed trunk. The vagus is both sensory and parasympathetic. And so we've been working on a method to specifically stimulate the parasympathetic nerves. And this is work that's been done by Ali Pincus in our laboratory. So she created mice that express a specific light-sensitive channel in the parasympathetic nervous system. And if you shine blue light over the trachea, just the parasympathetic nerves fire. And so you can see a mouse with blue light shining on it in this image. We know that the nerves are transfected with this light-sensitive channel, and that's what you're seeing in the left with the EYFP labeling. And if you expose those cells to light, they will fire, and that's what you're seeing on the right-hand side. So EYFP is positive. They have that channel. The little blue dash is when we turn the light on, and you can see that there's no firing of the cells if they do not express this light-sensitive channel. And this was work done by Ali with Sveta Adhikari at OHSU. We know that the light specifically stimulates parasympathetic nerves, and that's shown here. because if you turn the light on, you can increase that bronchoconstriction with physostigmine, which increases acetylcholine, and you can block it using atropine, which blocks muscarinic receptors. So we have a means now to specifically stimulate parasympathetic nerves. And at this point, I'm going to turn it over to David, who will be talking about the sensory nervous system.
Thanks, Allison. So the methods that we have allowed us to look at which nerves are responsible for hyperreactivity in different situations. As Allison said, Ali Pincus developed this method in our lab to use optogenetic stimulation to specifically stimulate the cholinergic parasympathetic nerves. For most of the data I'm going to present, we're going to look at sensory activation using serotonin. Although Ali has now developed a mouse that will optogenetically activate the sensory nerves. And we're going to look at these in two models of eosinophilic asthma. One is the IL-5 transgenic. This is the NJ1726 that Jamie Lee developed, in which IL-5 expression is driven by the epithelium-specific CC10 promoter, leading to a lot of eosinophils in the airways. And the other is an acute house dust mite sensitization and challenge. And in both models, we're going to look at reflex bronchoconstriction by stimulating the sensory nerves with serotonin, and then optogenetically stimulated bronchoconstriction, activating the cholinergic parasympathetic nerves. And we're also going to look at histologically at hyperinnervation. So when we look at these animals' response to serotonin in the panel on the left, you can see that both the house dust mite sensitized and challenged animals and the IL-5 transgenics are hyper-responsive to serotonin compared to wild-type saline-treated animals in black. So they look the same in terms of airway hyperreactivity. But when we look at specifically stimulation of the parasympathetic nerves using optogenetics, what you see is that the house dust mite animal is markedly hyper-responsive to stimulation of the parasympathetic nerves, but the IL-5 transgenics are not. So even though overall they're equivalently hyperreactive, the abnormality is clearly in the parasympathetic neurons in the house dust mite treated animals and not in the IL-5 transgenic animals. So potential mechanisms and these were data that Ali Pincus again developed, potential mechanisms of this may have to do with the location of the eosinophils or eosinophil state of activation or long-term effects of eosinophils. One thing that we know, as Allison mentioned, is that in the house dust mite treated animals, the eosinophils are closely associated with the parasympathetic nerves and are in position to block M2 receptors by releasing major basic protein. And we're just in the process of carefully analyzing the location of the eosinophils in the IL-5 transgenic mice. So it's possible that it has to do with the location of the eosinophils. But to get a little bit more at the mechanism of these differences, we're using a method that Greg Scott developed in our lab some years back, using tissue optical clearing and three-dimensional imaging. So the idea here is that you can take a lung, and that's a mouse lung in the before panel, which is opaque, remove the water and replace it with a material that has the same refractive index as the proteins in the tissue. And you wind up with a completely transparent piece of tissue, as you can see in the after picture there. You can then do confocal microscopy on a whole mount, so a very thick piece of tissue. We've done this with human airways, we do it with whole mouse lungs. And then we can use computer modeling of a three-dimensional reconstruction of the nerves to quantify the airway nerves. And this video is going to show you how that's done. So this is a mouse airway. At the top is the trachea. At the bottom, you can see the bifurcation into the two bronchi. And this is stained in green for airway nerves. And this is a patch of airway epithelial nerves, and you can see them running between the epithelial cells. And then here's a three-dimensional reconstruction of the nerves in this airway. And you can see how the nerves loop up and down between the epithelial cells. So these are sensory neurons in the epithelial layer. And then we're superimposing a computer model of the three-dimensional structure of the airway innervation. And once we have this model, we can use it to analyze the structure of the nerves in terms of branch points, total innervation, expression of receptors and neurotransmitters. And it's been a very versatile method for us. When we look at that in the IL-5 transgenic mice, what we find in the panel on the left is that there's about twice the epithelial sensory innervation in those transgenic mice compared to wild type. And that goes along with the hyperreactivity that you see in the graph on the right rather, where there are markedly increased response to serotonin compared to wild type. If we cut the vagus nerve in those animals, it largely eliminates that response to serotonin. So it's very clearly a vaguely mediated reflex bronchoconstriction. And that increase in sensory innervation increases that reflex. And that is pretty clearly associated with eosinophilia, because when we cross the IL-5 transgenic with the eosinophil deficient Phil mice, also from Jamie Lee, you markedly attenuate the hyperinnervation in the graph on the left, and you largely eliminate the hyperreactivity in the graph on the right. And this is from a paper done by Greg Scott and Matt Drake a few years ago. Well, what about in humans? Well, Matt undertook a study in collaboration with Rich Costello in Ireland, where they analyzed airway bronchoscopic biopsies from patients with asthma and use that same method. So what you're seeing in the middle here is a biopsy that's stained for PGP, the nerve-specific protein in green, substance P in yellow, eosinophils in pink, and then DAPI staining for nuclei in blue. And this is just a very brief video showing you that same method that we used in the mouse airways applied to human biopsies. And you can see not only the nerves, but also the association of eosinophils with nerves in this biopsy specimen. Well, when Matt analyzed those data, what he found was that in patients with severe asthma, and this is the graph on the left, the patients with severe persistent asthma had about twice the density of epithelial sensory nerves compared to both normal controlled subjects and patients with mild intermittent asthma. That tended to correlate with the eosinophil density in the epithelium, and that was measured by eosinophil peroxidase staining and voxel intensity. So there was clearly an association of eosinophilia with increased nerve density and with severity of asthma. So what we've shown so far is that eosinophils stimulate airway nerve growth, airway sensory nerve growth. There's increased sensory nerves in IL-5 overexpressing mice, increased reflex bronchoconstriction in those mice, increased airway sensory innervation in severe asthma in humans, and that's more pronounced in patients with eosinophilia. What about prenatally? Well, this project came from a study that was being done by Katie Liebold, and she was working with those IL-5 transgenic mice by breeding heterozygotes and using the wild-type littermates as controls. But when she looked at those wild-type littermates, it turns out that they looked much more like the IL-5 transgenics than they did like a wild-type. So the graph on the left here is looking at all wild-type offspring. But the ones in red are wild-type offspring of an IL-5 transgenic mother, and the ones in black are the ones that are wild-type offspring of a wild-type mother. So you can see that the ones with an IL-5 transgenic mother were very hyperreactive. And when Katie quantified the nerve length, again, those were about doubled in the offspring of the IL-5 transgenic mother. These mice had been born, there was no IL-5 left, there was no eosinophilia, but the nerve hyperinnervation persisted. And it turns out that the reason for that was that the IL-5 crosses the placenta and induces fetal eosinophilia in the wild-type offspring. And so if you look at these are all wild-type mice again, but with a mother that's IL-5 transgenic, the amniotic fluid IL-5 was very elevated. There was fetal eosinophilia in the blood, and the nerve length was markedly increased. And none of those changes, none of the changes in nerve length were seen when the father was a FIL mouse and the fetus was FIL. So it required not only IL-5 but also fetal eosinophilia to see that change in nerve development. After birth, the IL-5 was disappeared because the mother was no longer there to supply it. The eosinophilia disappeared, but the hyperinnervation persisted throughout life, along with airway hyperreactivity. Perhaps even more significant, when you take those wild-type offspring of an IL-5 transgenic mother and sensitize and challenge them with house dust mite, you get extremely severe airway hyperreactivity, so bad that the vast majority of those mice die from severe bronchoconstriction halfway through the serotonin dose response curve. And that was probably a result both of airway hyperinnervation and of an increased eosinophilic response in those animals. So in summary, eosinophil-nerve interactions cause airway hyperreactivity via multiple mechanisms. The parasympathetic nerve activity is increased by eosinophil major basic protein blocking inhibitory M2 receptors on the nerves. The sensory nerve growth is stimulated by eosinophils, including in utero, and persists after the resolution of eosinophilia. Hyperinnervation, when combined with acute eosinophilic response to antigen challenge, leads to very severe hyperreactivity. And the distinction between the baseline hyperreactivity in a hyperinnervated airway and the severe acute hyperreactivity after superimposed antigen challenge may explain the effects of IL-5 directed therapy in preventing exacerbations of asthma while having much less effect on airway hyperreactivity. And thank you very much for inviting us and for your attention.
Thank you, Allison and David, for the wonderful presentations. There's several questions, but I think for the sake of the time, I think I would like to ask you one question from the series of the questions. One question is about why MVP from eosinophils just blocked M2 receptor did not kill the nerves, and that's one question raised. Any speculations? Yeah.
Well, we first got into that because, and this was quite a long time ago. This is back in the 1990s. When you look at the structure of the M2 receptor, it's very heavily silated, and that gives it an overall negative charge. Other antagonists at the M2 receptor tended to be cationic, and because we were interested in eosinophils, we thought that perhaps one of the eosinophil positively charged proteins could interact with it. And it turns out that major basic protein does, with a KD of about 10 to the minus 5th molar. However, when we looked at other cationic proteins, we saw that eosinophil peroxidase was much weaker, and eosinophil cationic protein actually had no effect. So it's really not just the charge. There are other structural determinants that we frankly have not investigated.
Okay, I think we have a little more time, and there's one question. I'd like to just read out from the questions. Dear interactions between medications like SSRIs or gabapentin. Gabapentin. Yeah, so I'm totally out of the pharmacology with asthma or hyperreactivity to the antigen challenge. Do you have any
Well, I mean, it's an interesting question. It's not something we've looked at. Gabapentin is kind of a mild bronchodilator. SSRIs and other psychotropic medicines tend to have anticholinergic effects, which kind of complicate their effects in the airways. And we would predict that they would have some effect on this. On the other hand, there are better anticholinergics for treating asthma.
Mm-hmm. All right, thank you very much. I think we have to move on. Thank you very much again, Allison and David, for wonderful presentations.
Thank you so very much.
Thank you.
Thank you. So next speaker is Steve Ziegler. He's currently the director of academic affairs, and he is the director of the academic affairs of the Benaira Research Institute, and he is also the member in the Center for Fundamental Immunology. And he is also affiliated with University of Washington School of Medicine as a professor of immunology. As all of you know, he is a pioneer and leader in the FOXP3, regulatory T cells, as well as the TSLP. So I think today he is going to focus on the TSLP and talk about the biology of the cytokine TSLP. Dr. Ziegler, please come start your presentation.
Thanks, Hiro. And while we're getting started, what I thought I would do is thank the IES and it's especially Mark Rothenberg for the kind invitation for me to speak today, and for me to up front give a mea culpa that there's not going to be a lot of data presented here on eosinophils, so I guess I am the outlier here. I will present a little bit of data on it. It wasn't ours, but it's still relevant to the talk. And what I thought I would do is spend some time talking about TSLP in general in the beginning, and then focus on one area that we've very, very recently come across by accident on the role of TSLP in germinal center responses to type 2 in type 2 settings, because I think that might be relevant to some of the data that's present in some of the clinical trials with anti-TSLP biologics. And so with that introduction, what I'll do is I'll start for the 1 or 2 people in the audience who don't know what TSLP is. It is the most poorly named cytokine we have going. It stands for thymic stromal lymphopoietin, and that's because it was initially discovered by Andy Farr in the early 1990s as a factor that was secreted by a thymic stromal cell line. And it could support the growth of thymocytes and pre-B cells. And so Andy thought at the time that it was going to be the secret sauce for thymocyte development, and it turned out not to be. And so Andy dropped it, but then we started studying it for quite some time. And I was in there at the very beginning when I was at Immunex back in the early 90s, part of the team that cloned both the cytokine and the receptor from mice. We now know that TSLP is expressed primarily by epithelial cells at barrier surfaces, so the skin, the gut, and the lung have the highest level of expression. But it's also produced by other cells like fibroblasts, smooth muscle, dendritic cells at sites of type 2 inflammation. And it's now spreading into other forms of inflammation that I'll get to later. We also have found that the primary target for TSLP are myeloid-derived dendritic cells. And when they get exposed to TSLP, they then become competent to drive Th2 differentiation. But of course, lots of other cell types can respond. Basophils, mast cells, you can read that. And in fact, there are some papers in the literature suggesting that eosinophils can directly respond to TSLP, and they do have low level expression of the receptor. Now, the observation that basically completely changed the way we thought about this cytokine came from Young-Jun Liu's lab back in the early 2000s when he was still at DNAX. and it's actually Vasily Samuli, who was then a postdoc in his lab, who found this, showing that TSLP expression was massively elevated in lesional skin from patients with atopic dermatitis, and it was low or very baseline expression in uninvolved skin and in skin from healthy individuals. And this sort of became the seminal observation that linked TSLP to type 2 inflammatory diseases, and it's been extended to where TSLP is found in the lungs of asthmatics and in humans, and the noses of individuals with allergic rhinitis. And in both of those instances, the level of TSLP expression correlates quite nicely with disease severity. The receptor for TSLP is a heterodimer of the ligand binding chain, cleverly called the TSLP receptor, and it couples with the IL-7 receptor alpha chain to form a high affinity complex, which then signals through JAK2 and JAK1, as shown here, through STAT5 to do most of the biology. We haven't found any other pathways that are for sure linked directly to TSLP signaling, although there are some papers in the literature suggesting that PI3 kinase can get activated through TSLP, but my lab has not seen that. And so just to sort of give you an idea of how we think TSLP works, this could be in epithelium, it could be the skin, it could be in the airways, it could be in the gut. But as you all know, the epithelium is sort of like the first line of defense against what's going on in the outside world, and it gets insulted by a variety of agents, allergens, irritants of various kinds, pollution, diesel particles, bacteria, viruses. And what all of these insults have in common is that they induce the expression of TSLP quite rapidly. It's been referred to as an alarmin, along with IL-33 and IL-25. Basically, these are all cytokines made by epithelial cells that are made within a very short time of the insult and are around to basically inform the rest of the immune system in the vicinity that something has happened and something has to get done. And so, as I mentioned earlier, the prime responding cell is a dendritic cell, and these DCs, when they get a TSLP signal, can migrate to the draining lymph node and drive a Th2 differentiation, which then makes the same cytokines that can drive IgE levels and IL-5 to drive eosinophil recruitment. We also know that TSLP can act on basophils and mast cells to drive their function, to help to co-stimulate for cytokine production. And there are some new sort of mysterious pathways that TSLP engages in mast cells that are not quite clear, but it's been coming clear from some of the clinical trial data that I'll show momentarily. And we know that ILC2s can also directly respond to TSLP to proliferate and secrete cytokines. And so this sort of, canonical pathway is an operation in most type 2 inflammatory responses. And so what I'd like to do today is sort of segue into what I'm going to talk about by going into some very recent and extremely exciting data that's come out of a phase 3 clinical trial using an anti-TSLP antibody called tezepelumab. And this paper just came out very recently in the New England Journal, showing that tezepelumab, which is the anti-TSLP, in severe asthmatics with severe uncontrolled asthma, was successful at every benchmark for the trial, which included reduced exacerbations over a 1-year period, increased lung function by FEV1, et cetera, et cetera. And this was across the spectrum of patients. And for the eosinophil crowd, it included patients with blood eosinophil counts of greater than 300, between 150 and 300, and less than 150. And all of those groups responded quite impressively to tezepelumab. So right now, it's probably the only biologic that's hopefully will be approved shortly, within the next year or so, for uncontrolled asthma. And just so that my one piece of eosinophil data that I'll present today. One observation that's been clear from all of the trials where tezepelumab is used is there's a very rapid and very durable loss of circulating eosinophils. The reason behind this is not at all clear yet. It could be a direct effect on eosinophil, either viability, production, what have you, or it could be because the anti-TSLP is blocking, type 2 inflammatory responses, which would diminish the levels of IL-5, which is what happens. There are circulating levels of IL-5 are decreased as well in these patients, and there could be a sort of secondary effect of lower type 2 inflammation. But clearly, this is one of the clearest biomarkers for the effect of this drug in vivo in humans. Now, one other point that came out of this, and it's become, and this is relevant for what I'm going to talk about, is that individuals treated with this antibody also see a dramatic and persistent loss of circulating IgE. And we're not, at this point in time, we're not clear why this is. This is true from the Navigator trial, which is the phase 3 trial. It was true in patients from the phase 2 trials that preceded this. It's true in another trial where we just run jointly by Amgen and AstraZeneca, who are producing the tezepelumab with the immune tolerance network, in a trial called CATNIP, which was treating with tezepelumab in the presence of CAT immunotherapy, and those patients all showed a drop in IgE. And we're not sure exactly what's going on, but that's sort of a preamble to the data that I'm going to present in this talk, because this fits very nicely with what we've seen in the mice in mouse models for quite some time, which is mice that lack TSLP signaling. In this case, these are mice that lack the cytokine, show a dramatic loss of circulating IgE. And so these two pieces of data together suggest that TSLP is very important for driving IgE, which makes sense in many respects since it's a type 2 driving cytokine. But we wanted to try to understand whether this was a direct effect of TSLP on IgE production or sort of a secondary effect of TSLP being required for a sort of general type 2 inflammatory state. And so this is, the talk will be on TSLP and its role in the germinal center response. And all of the data that I'm going to present today is from a very talented postdoc, Phil Daumier, who joined my lab a couple years ago. It's what usually happens. I had no idea, about working on germinal centers. I swore I would never work on B cells in my lab. Phil comes into the lab, does one experiment that I'll show in a moment, and immediately it becomes the most interesting thing going. And this has all been a collaboration with Zia Rockman at Penn State Hershey College of Medicine. And it's not a coincidence that Phil did his PhD work with Zia, where he worked on germinal center responses. And so we've sort of teamed up together to work on this, and that's the data that I'll be presenting. And so what Phil did initially was, since we had these mice and he was trying to get a feel for what's going on in the lab, he took the TSLP deficient mice and just asked whether the lack of TSLP had any effect on steady state or spontaneous germinal centers in mice. So now these are germinal centers that form spontaneously in both mice and in humans. They're thought to be created for endogenous antigens, reactivated retroviruses, commensal bacteria, things like that. There are more of them in individuals who have autoimmune disease and in autoimmune prone mice. And what you can see here is a nice germinal center GL7 staining for the actual germinal center B cells surrounded by in a B cell follicle. In the TSLP deficient animals, these spontaneous germinal centers are both smaller and rarer. That's shown on the right side here, where the actual area of each germinal center is quite significantly smaller, and their numbers are also smaller, which again suggests that TSLP may be involved in sort of generically driving germinal center formation. And so we wanted to try to check this out more carefully by asking, first, a very simple question: is the two main cells that drive the germinal center reaction T follicular helper cells, or TFH cells, and germinal center B cells, do they express the TSLP receptor? And the answer is yes. So these are TFHs, and you can see they're marked by co-expression of PD1 and CXCR5, and they go through a quite elaborate differentiation pathway where the cells at the top, which had the highest level of PD1 and CXCR5 expression, are actually the TFH that are in the germinal center. And they actually have the highest level of expression of the TSLP receptor. And the red markers here are control, which is the same staining from mice that are knocked out for the TSLP receptor. The other cells that are in this pathway, have the receptor, but it's the most mature, most functional cells that have the highest level of expression. Likewise, germinal center B cells are marked by co-expression of GL7 and CD95 or FAS, and that's this red gate right here, and they have very high levels of the TSLP receptor as well, compared to the blue gate here, which are non-GC B cells, which express the receptor but not at the same level of GC B cells. So both cells that are involved in this process have the receptor, so they could be capable, and they both have the IL-7 receptor alpha chain as well. So they're capable of responding to the cytokine, but do they require response to TSLP for their function or for their existence? And so we looked at the various knockouts. These are TFHs. You can see wild-type mice have a nice population of these PD1 high, CXCR5 high cells. Both the cytokine knockout and receptor knockout have a dramatic reduction in both the frequency shown here and the number of TFH cells, suggesting that TSLP signaling is required for their steady-state existence. Likewise, germinal center B cells, which are in this gate here, are reduced in both the cytokine knockout and the TSLP receptor knockout, and that's shown for multiple mice here. And so this suggests that, it could be a very simple explanation. We know that one of the genes that's turned on downstream of the TSLP receptor is BCL2. So it could be very simply that TSLP is required for their survival. If they don't get that signal, they don't upregulate BCL2, they die. We've looked in both these populations in the knockouts to look at whether cells are dying. We don't see an increase in, say, annexin 5 positive cells. We don't see an increase in cell death. So we're not sure that that's the answer. We're still looking at this. I should have mentioned earlier on, this is all very recent data within the last year. It sort of goes into the category that I like to call not yet published data. Hopefully you'll be seeing it soon, but we're still trying to work out exactly what mechanistically is going on. And so that suggested that the cytokine was important for steady state levels of both TFH and GCB cells, but was it important for what germinal centers do, which is make antibodies? And so we tried to look at that directly. This is the model that we use. Take black 6 mice, immunize them with NP coupled to ovalbumin with alum as a adjuvant to drive a type 2 response. Now this is nice because there are reagents that allow us to directly look at NP specific B cells, so we can look at the response directly, and that's shown here, where we can gate on the GCB cell subset, and then look specifically with NP, in this case NP30, which is a reagent that allows us to see both high and low affinity B cells within that population. You can see a nice population in the wild type mice. In the TSLP receptor deficient mice, that goes down to almost nothing. And that's shown graphically here, where both total B cells, total GC B cells go down, and NP-specific B cells go down at about the same rate. And so TSLP seems to be important for generating antigen-specific B cells following immunization. And just looking at IgE expressing cells, both high affinity NP2, which is the number of NP molecules that are in the reagent, and NP26, I believe that is, or 25, I can't remember, but this is a reagent for low affinity. Both subsets of B cells following immunization are reduced in TSLP deficient animals. Again, suggesting that the TSLP response is important for not just the maintenance of germinal centers, but for their actual function. And I should mention that we think, I don't have time to show the data in this talk, but we think that the TSLP is actually being made by follicular dendritic cells, which are in the light zone of the germinal center and are important for this germinal center response. That's been shown in humans in patients with IgA nephropathy, and we've used both immunofluorescence and a TSLP reporter mouse that we got from Rich Loxley's lab to demonstrate that in mice. And so, we wanted to ask other aspects of the, at least the B cell side of the response, is TSLP involved in this? And in this case, we did the same experiment, collected at day 14 this time, and then just looked at TSLP receptor levels in various cells that are downstream of the germinal center, so plasmablasts, plasma cells, and memory. And plasma cells had the highest level of TSLP receptor expression that we found in any lymphocyte subset. And then when we looked at plasma cells in the deficient animals, we saw decreased levels of both total plasma cells, shown here, and in NP-specific plasma cells, which again suggests that it's not just the germinal center response, but it's the differentiation of cells following immunization that seems to be affected by TSLP. And what's interesting about plasma cell development is that it's quite known, at least from the transcription factor level, what drives plasma cells. So there's induction early on in the germinal center response of a transcription factor called IRF4, which can turn on BLIMP1, and it's this BLIMP1 BCL6 sort of counterbalancing that drives whether a B cell becomes a plasma cell or a memory cell. So high levels of IRF1 or IRF4 and BLIMP1 drive plasma cell differentiation, and BCL6 is important for memory cells. And so Phil looked at expression of IRF4 and BLIMP1 in these NP-specific B cells, and what he found, interestingly enough, is that wild-type mice and heterozygous mice had normal levels of this IRF4 positive, BLIMP1 positive cell that drives plasma cell differentiation, whereas the TSLP receptor deficient mice had almost none. And we've shown directly that TSLP signaling in these B cells can drive IRF4 expression. There's data in the literature suggesting the same thing in dendritic cells, and so at least on the B cell side of the ledger, it may well be that TSLP is functioning by inducing IRF4 early on in the response, and that then drives the response. And so we're still looking at that right now. So what about on the T cell side? Well, we wanted to do the same kinds of experiments, knock out the receptor specifically in TFH cells. And we decided to do this using a inducible CD4 Cree system. And that's simply because we and others, especially Warren Leonard's lab at the NIH, have shown that TSLP signaling in the thymus or in early T cell development does have an effect. And so we didn't want to have any confounding factors. And so the setup was to give tamoxifen by oral gavage 2 and 1 day before immunization to induce deletion of the TSLP receptor, and then do the same immunization with NP-OVA and alum. And what we find again is that these mice have a dramatic reduction in both high and low affinity and antigen-specific B cells. And so it suggests that TSLP is important on both sides of the ledger here, both on the T cell side and on the B cell side. We're not sure exactly what's going on in the T cells. We've started to look at that right now, but it suggests that it's an important, that this signaling pathway is a sort of a blanket important pathway for germinal center responses. And then just to close with what we think is the model, this is a germinal center, well, at least an artist's conception of what a germinal center looks like. And what I've shown you right now in this talk is that TSLP signaling is important both in the TFH side of things and in GCB cells. And we think that on the B cell side, it's through induction of IRF4 leading to plasma cell differentiation. On the T cell side, we're not sure. We don't think it's survival. We're looking at the ability of TSLP signaling pathway to induce CD40 and antibody affinity, and those experiments are going on right now. We're also looking in humans in the catnip study, where we see a reduction of IgE similar to the knockout mice, and we're looking at human TFHs. But we also have data that TSLP signaling is important earlier events in germinal centers, and we're now using a fate mapping system where we can tag B cells at various times during this pathway to ask where TSLP is important. And with that, I will close, and Hiro can ask me questions from the chat. Thank you very much.
Thank you very much, Steve. Wonderful presentation and very interesting data. I think we have I think we have studied the T cells and TSLP receptor for a long time, but we did not realize TFH and TSLP receptor side.
Yeah, it's good to find things by accident.
So Steve, there is one question in the chat about have you seen, is that if the patients are treated with anti-TSLP receptor antibodies, do we need to kind of concerned about kind of immunodeficiency in these patients, because TFH B cells interaction so important for antibody responses to the microbes as well? How do you think?
Right. So that's a very interesting question. We don't know right now. Obviously it's a relatively new drug, and there have been no adverse events in any of the trials that have been done. It seems to be extremely safe, but it's been also not been in people for very long. What the main question that we're trying to address right now is whether this is an effect of TSLP strictly in a type 2 setting, or whether it's a blanket functionality of TSLP in germinal center responses. And we don't know the answer to that. All of the data that I showed you in the immunized animals was using alum as an adjuvant, and we're now going back and repeating all of those studies using other adjuvants that drive a type 1 response, a type 17 response, etc., just to see whether it's a global effect or a very specific effect, and we just don't know the answer right now.
Right. Yeah, that's really the very interesting question to how global it is.
Exactly.
Maybe it's a Th2 specific responses that are kind of related to that. Did you have a chance to look at the TFH13 cells? Do they have any even higher expression of that TSLP receptor?
We're looking at that now, and we've just started collaborating with Stephanie Eisenbart's lab at Yale to do those studies, both in mice and in some human patients. So no data yet.
Yeah, very interesting. And there is a question in the Q and answer session. Just a moment. So does TSLP in germinal center contribute to antibody class switch? So I think we are talking about how the TSLP works in a type 2 response. Is that class switch?
Yeah, exactly. That's one of the questions we're trying to address right now, because it's not clear what the factor is that drives the switch from G1 to E.
Right.
And so we would predict that it could be TSLP based on the fact that, you know, IgE goes away in patients treated with tezepelumab, and the TSLP knockouts lack circulating IgE. The short answer is we don't know yet, but we are actively pursuing that.
Yeah, you are right on. I think we know IL-4 drive both IgG1 and IgE, but we don't still not know clearly about what driving from G1 to E.
Yeah.
So I think if TSLP provide additional signal to convert to that phenotype, that's great.
Yeah.
Good luck. Yeah. Okay, I think we have to kind of stop here.
Right.
And
Thanks,
thank you very much for wonderful presentation. Very interesting. Okay, thank you everybody. And I think we have time to close. And thanks again for all the speakers. And Florence, would you like to kind of close this seminar?
Yes, I would. Thank you very much, Hirohito. I have a few little reminders to make as well. So I'd like to thank everyone, first of all, for making this yet another great webinar. Thank you to all the speakers for very stimulating presentations. We had some excellent questions. I think we have more and more questions we should make this a longer and longer webinar, I think. Thank you also to Hirohito for moderating this session and keeping the time. I hope everyone appreciated the webinar. You'll soon receive an email with a link to an evaluation form, and if you could fill that in, it would be great so that we can further improve our meetings. Future meetings will continue to be programmed every last Wednesday of each month at the same time. So that's 9:30 a.m. to 11 a.m. Eastern Standard Time. The next one will be held on June 30th. Its title should tempt you to come join us again. It will be called Eosinophils and Parasites, More Than Just Effector Cells. Everyone deserves a summer break, so we've decided not to organize webinars in July and August, but we will be back in September for the trainee abstract session. We have decided to extend our submission deadline to June 15th and encourage you as trainees to submit your work, but also you as mentors to have your trainees participate. It'll be well worth your while because those who have presented the top-ranked abstracts will have their registration fee waived if they come to the next in-person symposium that is planned in 2022, probably in North America. This represents a value of practically $400. Details on how to format abstracts and how to submit them can be found on our website, as well as the way the actual abstract session will be organized. It'll actually be a mixture of selected oral presentations, and then there will be Twitter-based poster presentations. With this, I'll close the meeting, and I wish you all a good day. Bye-bye.
Bye-bye. Thank you. Thank you, everybody.